Okay, so anyway, the simple title of this is stem cells. Of course, we're mostly from the Cell Surgical Network looking at adipose stromal vascular fraction, or what we call SVF. But I assure you, we are in a new era of medicine. I don't know how many people have thought about it, but everything we've been doing in medicine up till now is to take care of your cells. And now we have actually a way of moving forward with cell therapy. I like the term CRT, cell renewal therapy. In fact, the R could be cell replacement, cell repair, cell regeneration, whatever you want. CRT is really going to be the new era of medicine. I think stem cells, and especially personalized medicine, is going to be what we're going to be looking at for everybody all over the planet. Hey, Sean, we got to get these slides to advance here. All right, here we go. First, couple simple disclosures. I'm part of the California Stem Cell Treatment Center. We also, the Cell Surgical Network, we have a program, Cells on Ice, for growing your cells. But I'm also involved in China, and I'll talk to you a little bit about that as we move forward. A lot of you don't know who I am, but my original training was in otolaryngology, board certified as a head and neck surgeon, but also have spent the last 30+ years as a cosmetic surgeon. In fact, I was the past president of the American Academy of Cosmetic Surgery back in 2010. I'm a fellow American College of Surgeons, and currently I serve as a co-medical director and co-founder of the California Stem Cell Treatment Center Cell Surgical Network, along with my colleague, my partner, Dr. Elliot Lander. Many of you have heard Dr. Lander speak at other meetings, and Dr. Lander's background is a urologist. Kind of interesting that a cosmetic surgeon and urologist should come together, but it's been a really nice union. I've enjoyed working with Elliot this last 6 years. Now, I'd like to start out with the punchline. Here's what I think. Stem cells are investigational. They're not really experimental. These cells have been in our body since the beginning of time, and we've either gotten better with these cells or didn't get better. And I think what we're doing now is bringing back the oldest form of medicine, not just the newest form of medicine. Our bodies are made up of nothing but cells, maybe 40+ trillion cells. And so if you think about it, as we age, our cells start dying. Now, potentially, we have replacement parts in the form of cells. That's why I think CRT is going to be so important, because we can actually start thinking about replacing our parts, maybe even increasing our longevity substantially. There was a good article in the Wall Street Journal written back in October, and Matt Ridley wrote a whole book about this, but the effect was the steam engine came long before the science of thermodynamics. In other words, innovation starts with people like ourselves working in labs or working in clinics, and then the laboratory, the PhDs, they come along and they figure out why things work and maybe even make it better. But a lot of times innovation starts in areas you least expect it. I think we're in a transition in medicine right now. Everything we've done up to this point is how do we care for ourselves, and that may be diet, exercise, good night's sleep, supplementations, hormone replacement. But going forward, we have replacement parts. Now, they've already been used in the form of organ transplantation, but now we can use them in the form of cells. Bone marrow was one of the most popular original stem cell replacement parts. It still is. We've looked at embryonic cells and other forms of cells, iPS cells, induced pluripotent cells, things like that. But I think fat is pretty magical. It's amazing. Maybe it's just dumb luck, but your fat is loaded with stem cells, and it's a very simple thing for us to use. A while ago, Elliot said, you've got to read this book, The Black Swan. It's his favorite book. Nassim Taleb wrote this book and talked about the idea that black swan's a pretty rare bird, but it's a real bird. And it talks really about disruptive technologies, the idea that something can come along, like the iPhone, for example, and it just changes so much about technology and about the way we integrate in society with different functions and different organizations. But how are we a new technology in the stem cell arena? I used to think you know, there's somebody going to come along with cells in a bottle, and that's going to replace us. It'll be allogenic cells, but it may be that we're actually the black swan. Anybody doing personalized regenerative medicine may be a black swan in the sense that we have the ability to make these cells in a very inexpensive way. Fundamentally, if we can put a plug in some place in anywhere in the world, in the middle of Africa, we can put in our centrifuge and our incubator and literally make somebody their own stromal vascular fraction and ultimately give them their own stem cell therapy. I'd like to look back at the early history, and I'll explain how we started a little bit. Yves Allouz and Pierre Fournier were two of the dynamic fathers of liposuction, and even more importantly, lipotransfer. So these guys came along and said, hey, you can take the fat, inject it in here, voila, you got a lump. And we started doing lipotransfer. Back in the 80s, we had a little tea strainer. We would collect the fat, strain it out, put it in another syringe, and re-inject it through these Coleman cannulas. And that was pretty cool. We tried other techniques like the Lipovox tulip system, and that seemed better. And ultimately, I met Dr. Lee. Back in, I think it was April 2008, Dr. Lee and his contingency came to my offices and introduced me to an invention of his. Dr. Lee is like the Tony Stark, remember, from Iron Man, of medicine. This guy's a plastic surgeon, but he'll invent anything. And he's done some really amazing things. He started out with the lipo kit, which was a big box, which was a centrifuge and a liposuction machine inside of it, and we could harvest the fat in these specialized syringes. And what you see here is the grease separated on top of a weighted piston that's filtered, and then you have the compressed fat and then local anesthesia that you get rid of. And this compressed fat was much better, and in fact it's what we use today for our facial fat grafting procedures and other fat procedures. Also, the system was relatively closed, so we could transfer fat into these 1 cc syringes and inject it through a little tiny 0.9 millimeter tulip cannula without making a big mess. If any of you have done fat grafting other ways, you know it can be real greasy and it gets to be very messy. On top of it, I started out using this stuff just for facial rejuvenation and other defects. Fundamentally, the thing I've noticed about using this, it just made the system much easier, much more reproducible, and for me, in my hands, I was getting really good results. So I would have these patients, we could fill up their face, restore them in a more natural way. I've talked about this for years, but I believe we've completely misdiagnosed aging. Aging is, again, the loss of cell volume. Predominantly, your fat cells live 7 to 10 years, and as they start to age, our face starts to disintegrate in a sense. Our skin looks like it's sagging, when really it's somewhat imploding. We're losing the anterior dimensions of our face, and by putting fat back in the face, you can restore those anterior dimensions, and we can get some pretty good results. But while I was doing this cosmetic stuff, I really wasn't thinking about the idea that what I was doing was a stem cell fat transfer. And we understand that now. We can fix a lot of these defects very nicely with adipose tissue, but now we understand how it works. And the real key is the stem cell. In fact, Kotaro Yoshimura really did a great job in delineating this stuff. Yoshimura arguably is one of the leading authorities on stem cell and plastic surgery combined. He popularized the CAL system, which we'll mention shortly, which is cell-assisted lipotransfer. But Yoshimura also showed that when you inject fat into a face, a lot of the fat cells die because they need oxygen. The stem cells can live a much longer time, and they end up replacing the fat cells. So that was a very neat discovery of his. So what is a stem cell? Most of you, I'm sure, know what it is, but think of the stem cell basically as an unspecialized cell with all your DNA. It hasn't become the specialized cell yet. Fundamentally, stem cells can replicate and they can differentiate. They can turn into the tissue that we need them or want them to become. Embryonic stem cells are commonly thought of as the main source of stem cells. In fact, there's over 261 strains of embryonic stem cells being evaluated even today. There are moral issues with them, and they are sometimes hard to keep from turning into tumor cells. They are what we call pluripotent or omnipotent, a blastocyst. Those initial stem cells from embryo can turn into every part of a body. But today we found that we actually have a lot of adult stem cells. In fact, they're all over the body. Almost all of our body parts starts with spare parts, which are essentially the stem cell. A lot of people will call them progenitor cells because they believe they're destined to become whatever that next cell is going to be. But these are your own adult stem cells, or what we call autologous. They're from your own body. The one we started working with the longest was bone marrow. People have all heard of bone marrow transplant, especially for leukemia, things like that. And a lot of the early work, especially with therapeutic orthopedics, was done by people specializing with bone marrow. In fact, Chris Centeno is considered one of the pioneers, I would say, in bone marrow therapy. And he's done a lot to move that part of the industry forward, if you will. But there's some other issues with bone marrow, and that led us into looking at fat. When Dr. Lee came to my office and told me that, by the way, you can not only use this equipment to make fat that's better for fat grafting, you can also make stem cells out of it. I'm going stem cell, schmem cell. I really didn't understand. But between 2008 and 2010, I did a lot of research, and I realized that I was missing something that could be very important. So our stem cells, particularly from adipose and bone marrow, often referred to as mesenchymal stem cells. Mesenchyme is basically the support structures of your body. So you think of those as cells that can turn into bone, cartilage, muscle, nerve, fat, connective tissue, things like that. Fundamentally, we've got research that my colleague Jackie C. and his associate Dennis Ling did, and showed SVF, the cells from fat, could be turned into practically every cell in your body. The simple concept, the way I like to look at it is very simple-minded. A lot of people think stem cells do a lot more than just turn into new cells, but fundamentally, a stem cell in fat, which may be a pre-adipocyte that's going to turn into a fat cell, as well as the stem cells in fat are in the lining of the blood vessels, and some are even on top of the blood vessels. Think of these like an envelope with all your DNA, and they just have a different address on it. And what really makes that stem cell special is the signal it gets. These signals we call cytokines. They also, you'll hear the term growth factor or exosomes. They may be little bits of messenger RNA that will ultimately connect with the corresponding part of the DNA to tell it what it needs to become. We were able to get this slide that is not mine originally, and I would love to give credit to the author, but this is a zebrafish with an induced wound. You can see the wound at the top is excreting a growth factor called SDF1, and then you see a blood vessel at the bottom. And what you're going to notice are all these little cells that go sliding up to the area of the wound. And this is almost like, look like sperm going to an egg. But fundamentally, your wound secretes cytokines. These signals are what call the stem cells to action. When you have an injury in your body, these signals can call your cells to action. And unfortunately, what happens many times is instead of having stem cells go to the area where you can actually repair the area with like tissue, you end up getting a lot of white blood cells going to that area, and you get scar tissue instead. Well, scar tissue is good for healing a lot of different wounds, but it's not great for repairing things like arthritis and other things. Like if you had a heart attack, you want to grow new heart muscles. You don't want to grow a scar, which is what usually happens after a heart attack. And so we looked at the idea of using fat to repair a lot of these degenerative conditions. Why would you use fat instead of bone marrow? Turns out that fat cells, 1 ounce of fat could have anywhere from 5 to 30 million stem cells, while we could take out 3 times as much bone marrow and get maybe 150,000 stem cells with some of the new technology. At any rate, the same cells in bone marrow are pretty much, the cells in bone marrow are pretty much the same as they are in fat. We've got plenty of studies to support that. And we've got people out there like Bob Harmon and his company with VetStem. They've treated well over 11,000 animals, and they may be up to 12,000 by now. He's done everything from little toy poodles to big rhinoceros and giraffes. It's unbelievable what they've done. But they've used fat-derived cells. They process the cells, send them back to the local veterinarian, and treat these animals. There were reports out of Europe, all over the place. And for a while, China was doing a lot of stem cell work. A lot of countries around the world have pulled back on stem cell work because they're afraid that the SVF is controlling us and don't want us to be doing this work. Interestingly, Japan now has opened up a registry, and they're doing a lot of stem cell work, as is other places like Korea. In April 2010, I had a neat trip to Japan. This is my wife during the sakura season, cherry blossom time. And I got to visit with some really good plastic surgeons. In particular, I spent a lot of time with Kamakura and Yoshimura. Yamakawa became a good friend of mine later on. He originally started with Kamakura, did a lot of breast augmentation with adipose-derived cells enhanced with you know, the stem cell or the SVF. But Yoshimura really spent a lot of time with us and explained how the stem cells seem to work. And I think that's where I learned the most. Interesting, Kamakura had 2 Satori machines. If any of you know anything about the Satori machine, it's an automated machine. They cost about $100,000. I heard they're selling them in Japan now for like $300,000 with about $2,500 in disposable parts. And he was using these machines to harvest fat and then process some of the fat into SVF, or stromal vascular fraction, augment the fat, and then use it for breast augmentation. When I left Japan, I came home, I said, this is ridiculous. We're wasting stem cells to make bigger breasts. I mean, it's not the worst thing in the world. Your adipose has literally millions of cells in it, and some people figure that when you're in your 20s, you may have 500,000 to 600,000 stem cells per cc. So it kind of made sense to take some of those stem cells, add to the fat, and you would get better survival of your fat cells. A fat cell will live about 7 to 10 years. That's why we get older looking. I'm older looking now because I've lost a boatload of fat in my face. But if I could inject a little bit of fat in my face, I can restore the natural contours of my face and look younger. So this is what we're working with. When we do fat grafting, as Yoshimura showed, you're putting a graft into a low oxygenated area. A lot of the fat cells end up dying, but their death stimulates the stem cells to replace them, and the stem cells often replace most of the fat cells as well as grow blood supply for that graft. This is a basic idea of taking those cells, adding it to fat so you have an enriched graft, and you do things like this. I've even done it too. You get breast augmentation. They look pretty good, last pretty well. But you need a lot of fat just to make a small change in a breast. So when I look at the whole picture, I say, wow, we could do all this stuff cosmetically, but therapeutically, it's off the charts. And why are we wasting our fat just making breasts bigger when we can do all this therapeutic stuff? I was president of the Academy of Cosmetic Surgery back in 2010, and the last thing I wanted to do was get in trouble with the medical community by doing some of this early work on knees and hips without an orthopedic surgeon. Dr. Tom Grogan eventually agreed to see patients with me. And you know what? As a beginning doctor, if you're interested in doing this, I implore you to set up these multidisciplinary teams. It is so much fun to be back in medicine, working with groups of doctors, besides just going to my office, seeing my patients, and going home. I actually feel kind of like, in fact, I was talking to one of my colleagues today, and she said to me, says, you sound like a new resident just fresh out of practice or starting practice. And I almost feel the same way. I mean, I've been in practice over 30 years. I'm having more fun than ever. But a lot of the fun is working with different colleagues from different specialties. Any of us who are in medicine, if you really know that, you know, if you're like me, you enjoy medicine. Most of us really enjoy our ability to practice medicine. Anyway, Dr. Grogan said he would see my patients. Where do you get patients? Well, most of my patients come in aging face. If somebody's older, oftentimes they have arthritis. So when I started telling people how we could put fat in their face and it had stem cells in it, they said, gee, I wish I could use that on my knees. Next thing I know, I'm turning a $20,000 facelift into a free, you know, stem cell procedure or SVF procedure. But it paid off because a lot of these patients did not have to get their knees replaced, and you know, 6 years, 7 years later, they're still walking around without pain. So we would see stuff like this routinely, where we would, this is a picture you see in the back of the airplane where you have almost like bone on bone, but there's some cartilage in there. As long as there's some cartilage, you can get a signal, and eventually you get some improvement. And you can see there's a joint space that you can appreciate. And here was another example 3 months later. And these people were coming back and say, hey, my pain's gone. And instead of being gone for, a week or 2 weeks or even a month or two, it was gone for months and even several years. Again, here's another example 3 years later, a very narrow space, and now it's opened up. And these patients, more importantly, come back and say, hey, my pain's gone, I feel better. So how does this work? Well, basically, as arthritic cartilage breaks down, you give off these cytokines. What we usually get in orthopedics are these severe cases, or even almost bone on bone. And what we're learning now is if you can get them early on, more acute stages, you have a better chance of fixing it. If you think about it, most people come to us because they think this is a new technology. We have no idea what we're doing. We may be the fringe doctors on the fringe of medicine. And so they're going to take a chance with us because the next step, instead of getting all the traditional treatments like NSAIDs and steroids and hyaluronins and maybe arthroscopy, the next step is a total joint replacement. And they're willing to try something as radical as their own stem cells before they do that. The interesting thing is, if we get them earlier, we do a lot better because the signal's louder. In other words, the cells work better around a lot of cytokines. So after having some success with Dr. Grogan, I told you Dr. Lander and I teamed up, and Elliot's a urologist. He's really well known. He speaks all over the world, and he's got offices in the desert. And so together we set up an office in the desert, and then we also have one in Beverly Hills. And then Jackie C., who's an expert cardiologist, joined our group. And actually, Jackie has experience in stem cells going all the way back to Kazakhstan many years ago. I already told you about Bob Harmon and his work with VetStem. Bob came to me because he had a bad shoulder, but on top of that, he brought a scientist with me, to a laboratory assistant, and they evaluated everything we were doing, and he actually helped us to modify our program to really close it down as best we could and improve it. We formed a multidisciplinary team. We have neurosurgeon, cardiologists, interventional radiologists, a whole bunch of different groups within our group. Eventually, by talking about this at some of our meetings, people would come up and said, hey, can we join your network? Well, one of the first things Ellie and I did is realize we needed a protocol that was IRB approved so we could publish our data. We knew this was investigational. At this point in time, I don't even think stem cell work is experimental. We know what to expect from using stem cells in the right conditions, but we need to investigate so we get the numbers and the data. And so that's what we did. We started with IRB approved studies. We really looked at doing safety protocol first, and we built a huge database online, which we're hoping we're going to be able to provide to other doctors outside our network, or maybe expand the cell surgical network to other doctors. And we actually are working with the FDA. People think we're afraid of the FDA. We're hiding from the FDA. We've been told we're going to be thrown out of town by a countless number of doctors and PhDs and all kinds of people who like to blog about us. But we work with the FDA. We actually have an IDE application, that means investigational device exemption application pending, that we're working on with the FDA right now. We're almost at the approval stage. And on top of that, we talk with the FDA regularly to let them know what we're doing, what we're up to. We've got a lot of people who've joined our network. There's a lot of great people in the network. But what makes the network particularly fun is everybody's getting the same results we are. And whether you do our technique, which is basically in surgery, in the surgery room technique, or you use a laboratory technique, anybody working with adipose tissue and these stem cells, even bone marrow, can tell you they get some pretty amazing results. And at this point in our career, we expect them. Even though we get them, we're still amazed. We have our colleagues like this Dr. Lou Obi, who's given meetings. He's in Jacksonville, Florida. He's a renowned plastic surgeon. And a lot of people now are joining our group around the world. We even have a team in China. I started out in Shanghai, China several years ago. And this was the place I started working. It was called WA Optimum Healthcare. And I was asked to go there as their cosmetic director. And when I started in 2011, I just started doing the work with the stromal vascular fraction. And in fact, one of my first patients was the wife the head of our stem cell research institute that we have, which is now called the Cell Biomedical Group and trades on the NASDAQ as CBMG. So it's kind of a small world the way everything's worked out. Eventually, I went from working at WA and I got involved with this gentleman on the left-hand side of your screen, Benno Zhao. I was introduced to him by the gentleman on the right-hand side of the screen, Steve Schweller. And Benno had a little company in Suzhou, China, which is about 60 miles west of Shanghai, where they were banking and expanding stem cells from adipose tissue. But they didn't have a point-of-care system. So we teamed together, we had a nice little peanut butter and jelly connection, and we started setting up point-of-care systems throughout China. China got into a situation where they were the Wild West and everybody was doing stem cells, and then they shut down every stem cell clinic around China. And so we started going to party officials, discussing this with them and having a meeting with different hospitals, public and private hospitals, until we got approval to start setting up some of these clinics. We got involved with some amazing people. I'll tell you a little bit about Dr. Zhu. There's a lady on the right-hand side of the screen holding her certificate, but she heads the International Spinal Cord Injury Treatment Center in Kunming, China. And they're members of our cell surgical network and part of that RCC ReStem team. They're using cells to repair paralysis patients right now. And it's amazing stuff. I'll show you that as we go along. Anyway, I got to meet a whole bunch of people throughout different areas in China. And right now, I'll tell you, there's a city in southern China called Chongqing. And the Chinese government, and about 6 months ago, pulled the plug again on stem cells because somebody died from natural killer cell infusion, and they decided that nobody should be doing stem cells unless it's approved in a public facility. Well, the Chongqing party officials are working with us, and now we're on the verge of getting this going again through their public and private hospitals that they control. Once that gets going in the next few weeks, we'll be able to resume all the treatments that we're doing throughout China. And so this is a really exciting time for us. And this is just a view of Chongqing. Chongqing is one of the biggest cities in China. I'd never been there until about a year ago. There's over 28 million people there, pretty much the same size as Shanghai and Beijing. So the system we use today now is a smaller version of that original lipo kit you saw. We call it the CSN time machine. It's kind of a fun name for what we're doing, but in many ways it makes sense. We're technically reversing the clock on a lot of patients. And so it's basically a small centrifuge, and this is very portable. It weighs about 25, 30 pounds, and a small incubator, and that weighs maybe 10, 15 pounds. So if you really need to drive it anywhere, you easily can. But it requires 25 to 50 cc's of lipoaspirate, and it's a nearly closed surgical procedure. I say nearly because no surgical procedure is actually closed. We work in an environment of air. The ambience has particles in it, and we're cutting through people, and therefore it's not completely closed. But, and this is what I just said, no matter what we do, when you're doing point-of-care procedures, it's never completely closed. The process we do is a mini liposuction. It's done with local anesthesia only. It takes 5 to 10 minutes to do, and it is not with tumescent anesthesia. The important thing is we're blocking right under the dermatome plexus, so we block all the nerves going down to the fat without having put a lot of local anesthesia in the fat, which can be lytotoxic. And by the way, a lot of people will ask, gee, I had adipose done and it didn't work for me. But most, I would say 90% of the doctors doing adipose harvesting are using tumescent anesthesia, which fills up the fat with local anesthesia and may be cytotoxic. In other words, may actually kill the stem cells. And we've seen evidence of this happening. Anyway, we use an enzyme collagenase to separate the collagen that binds the cells together. It doesn't really affect the cells, it just breaks down the collagen. And basically we just wash and filter out the cells so we can deploy the cells into any joint in the body, but we can also give it through an intravenous deployment or even directly into the brain through an Ommaya reservoir. And I'll show you some of that stuff. Basically, that's it. We harvest the fat with this neat syringe technique. We have a very simple set of equipment, And ultimately, we filter all the cells through a 100-micron filter, so there's no chance of any embolizations when we deploy it. We can count the cells. We have more sophisticated counters than this, but fundamentally, we exclude all the small red blood cells. We count the cells that are larger than 10 microns, because your stem cells are like 13 to 17 microns in size. Red blood cells like 6 to 8 microns. So we count the larger cells, and we can look for cells that are viable. In fact, we even take samples of these, and we send to the lab, and we can show that your stromal vascular fraction is loaded with CD45 and 34, which is hemopoietic and stromal-vascular, or adipose-derived cells, or mesenchymal stem cells. In fact, we will look at many patients and see they all have stem cells. Some have high quantities, some have lower quantities, but they all have some stem cells. The clinical uses are all over the place. Fundamentally, these cells can be used for any cell injury in the body. So if we start making claims with the FDA and people want to know, is this FDA approved? We go, God, they're going to be approved for every use in the body. If you had pneumonia, you would want to use stem cells to repair your lungs. And we don't, right now, we tell people don't use this if somebody has an acute infection. But fundamentally, it'd probably be a good thing to do. The big question initially was, is it safe? And we've addressed that pretty well. Certainly, we know we're doing a nearly closed system because after we harvest the fat does not contact the environment at all. But the most important thing is, with your patients, make sure they don't have any disease in their body, particularly dental issues. And so the real issue with the FDA is they want to make sure we prevent communicable disease. We can't completely control whether our patients have possible disease in their body. We're cutting through skin. There could be bacteria there. But what's neat to know is we haven't had any infections in any of our patients. We've had no medical malpractice occurrences, and we have real good safety data, and I'll show you some of that. In fact, we just had a peer-reviewed paper published a couple weeks ago, finally. I mean, we've been working on this a long time. We hired a few PhDs to help us really hammer it out. But it's a good prospective study, and it looks at over 1,500 patients. And of those 1,500 patients, 1,477 actually had IV infusions. And even with those IV infusions, they had no problems. No tumors, no emboli, no real serious adverse events directly related to the stromal vascular fraction. And these are really good things to know. By the way, we've been doing liposuction and lipotransfer for over 30 years. So I may have mentioned it earlier, but just to reiterate, we know it's safe because I've been sticking fat in people's faces with free stem cells for well over 30 years. And this has been done all over the planet, and there haven't been any unusual or interesting reports that you would think would come out if free stem cells were doing anything dastardly to our patients. Our clinical trends are pretty impressive. Orthopedic cases do real well. They seem to be the low-hanging fruit and easiest to treat. But we've got about 60 to 70% improvement across the board for most other conditions. This just shows orthopedic conditions in the 80-85 percentile for improvement straight across the board. Here's an example of knee pain. This is stuff we can do with our database. We can pull out examples of people at rest, standing, walking, running, and you can basically see that there's a good decline in most knee pain. Running, not so much, but certainly at rest and walking. And that's really substantial even after 2 years. If you look at other conditions, autoimmune, pulmonary, cardiac, neurodegenerative conditions, and then a bunch of urologic conditions, we have pretty good improvement. We're doing things to get better at it. Neurodegenerative conditions are very difficult. They're probably going to take a lot more than just stromal vascular fraction. Although we've had some good improvement with stromal vascular fraction used over and over again, but now when we're going to get to the point where we can bank our cells and expand it and get large numbers, I think we're going to see some difference. We've had a lot of articles written about us, a lot of things said about us. Sometimes it's not always, you know, the nicest stuff. But of course, whenever you start with something, people always love to criticize you. But we've had some good articles about us, even in Popular Science. The guy who wrote the article in Popular Science was actually became a patient. he wrote the original article about our other patients, and then one day he calls me up and says, hey, you know, I've had this back problem. I've had multiple treatments on it. I've never gotten rid of it. It's been killing me for like the last 4 or 5 years. I like living in pain all the time. I said, well, you know, we could take a shot at it. Sure enough, we gave him SVF treatment. His back pain cleared up, and the article became a first-person article. You know, there's a German philosopher, Arthur Schopenhauer, who said all truth passes through 3 phases. And I think a lot of you have heard this. We rarely give Schopenhauer credit for it, but I guess he deserves it. Not that he cares at this point in his career, but, you know, first it's ridiculed, next is violently opposed, and then eventually it's accepted as having been self-evident. I think a lot of us should be at that self-evident stage, but most of us are at the violently opposed stage right now. People are still ridiculing us, and it cracks me up every time I hear somebody say, yeah, I went to my orthopedic surgeon, and they said this is just voodoo medicine. And it's kind of funny at this point to hear stuff like that. Dr. Lander and I wrote this book, Stem Cell Revolution, which kind of gives a view of what we went through starting this program up, our approach to it, and then some interesting stories about our patients. The other criticism when we started was this could be just a placebo effect. But you know what? If you look at some of the 11,000 animals treated, well, they could have placebo, I suppose, but a lot of our patients have had multiple treatments and they failed all of them. If they failed their multiple treatments, they clearly didn't have a placebo effect from those treatments. So it's a little bit illogical to think that they get a placebo effect from SVF when they couldn't get it from anything else. So orthopedics is obviously a low-hanging fruit. We've talked about that. People want to know how quick and how long it works. A lot of people are frustrated. Like shoulders, for example, could take up to 3 months before they get better. And I had shoulder patients just come in very upset that after a few weeks they weren't any better. And then I thought, well, I'm going to lose them. And they'd come back 3 or 4 months later and go, hey, my shoulder pain's gone. And I said, well, maybe it's from something else. They go, no, I've tried everything else. It just took a long time and it finally went away. I actually had stem cell treatment for the first time for a sciatica that I had. And I've seen back patients get better almost overnight or within a week or so. And my sciatica completely cleared up, but it took me 4 weeks. You could do some neat radiology. And one of our partners, Dr. John Feller, who's a great radiologist, talks about using gadolinium so you can isolate hot spots, and then actually using CT guidance to put the cells right on the spot. You can do that. In fact, this is my my CT showing the cells going right in my facet, and here's one showing cells going into epidural space. He puts a little air in there to show the epidural space and then puts the cells in. And you can do that, but you could probably put the cells in close to the injury and still get a good effect because the cells will seek out the cytokines that are calling them. The louder the signal, the better they'll do. So we have a lot of IRB approved studies right now. There's about 10 or 12 of them. We're looking at different outcomes for all kinds of things from knees to cardiac to neurologic, including an Ommaya reservoir procedure we're doing. We've had some good results in Parkinson's with just intravenous injections. In fact, one of them was pretty amazing. You'll find a lot of these stories all over the place. A lot of them are quite anecdotal, but when you see people who can barely walk, can not stay up, who can't do their tasks, such as this composer I once treated who couldn't play piano. Now he's back playing piano and composing. It's pretty remarkable. You can do some objective studies like this and show people are less shaky over time. And then there's things like our patient with myasthenia gravis who couldn't ski for the last 4 years, and now she's skiing again. Multiple sclerosis. We have dozens and dozens of MS patients. And a lot of them right now we're working on the Ommaya program, and we're actually seeing some pretty good results. And I'll talk to you about the Ommaya in just a minute or two. Muscular dystrophy, it seems to work real well for that. RSD works well. CP, who would think it would work well in CP? But think about it, cerebral palsy fundamentally is a vascular disease where you've injured the brain and you get a vascular compromise, and so you probably get the same problems you get with a concussion or any of these injuries where there's vascular compromise and also an inflammatory condition secondary to it. but CP seems to respond pretty well. This patient, Julia, is a little bit chubby here. Why? Because she was on a boatload of steroids for optic neuritis. We met her. She was legally blind. She could see pinpoint white light, and that was it, on 40 milligrams of prednisone. And she was at the point she wanted to go off her prednisone and be blind instead. We've given her over 30 treatments, but she stabilized. And she was able to pass her driver's test and get off all her prednisone. And for over a year now, in a couple months, she hasn't been back because she's been stable. So we must have caught up with this. This is probably a variant of multiple sclerosis. And these people, you know, they kill you because, like, this is a little Christmas card from her daughter thanking us for getting her mom a second chance. Stuff like that is amazing. COPD sometimes reacts really well. We've even had people do well with pulmonary fibrosis. We had several patients with cardiac conditions do real well. We had one guy on Stanford transplant list waiting for a new heart because he had an 18 ejection fraction. We got him up to 38, and he was able to get back into his routine activities, and they took him off the list, of course. Dr. Landers worked on some amazing neurologic stuff. To me, the interstitial cystitis is the neatest thing because these people are in horrible pain. You know, they have frequency, painful urination, and Dr. Landers is able to help at least 65% of these people. They resume their lifestyles. He's done some real good work with Peyronie's, and he's been doing some neat work with erectile dysfunction and Peyronie's using the a shockwave device by Storr. It's called the Deactor. And that's a pretty neat way of breaking up the blood vessels and getting the stem cells to increase blood flow. Now I'm going to talk real quickly about Dr. Duma and the work he's doing with the Ommaya Reservoir. He recently got featured in a front page article. The Ommaya Reservoir is a little port that goes directly into the ventricles, and we can put cells right into the cerebral spinal fluid. We had to do a little rat study first to show it was safe. These little guys did quite well and showed that the cells got there. This shows Dr. Duma just putting the cells into the port. So you go in and you can remove some of the cerebral spinal fluid. And then what's really neat is then you can just go ahead and inject, here I'll show you, the SVF right in, and you flush it out with some more cerebral spinal fluid. Pretty neat little gadget. We've talked about this all over the world, including in China, and here Dr. Duma is talking with John Gurdon, who was a guy who did the first vertebrate clone. This was a front page story that just appeared a few days ago on Dr. Duma's patient who had Alzheimer's. What's really amazing about this is Duma was able to take start giving him SVF, and then he started giving him expanded cells, I believe, and he was doing the work in Dr. Landers' office. They measured the hippocampus. Initially, it was at 5 percentile. At 1 year, it was at 28 percentile, and at 2 years, it was at 48 percentile, which is considered normal. His cognition increased, as did the hippocampus, which is the memory center in the brain. And this is actually probably the first time you'll see actual objective evidence of the brain growing back and somebody improving with Alzheimer's. Pretty amazing stuff. We also did a little pilot study with autism. We used the primary relative to use their cells with SVF, and we actually have some good results in 4 patients. We're working with another gentleman, Dr. Aladar Salai, in a cancer study. They've developed oncolytic viruses, and we found that if we can incubate them in the stem cells, we can deliver them like a Trojan horse. This will take the virus to the cancer, the virus jumps into the cancer, grows, kills the cancer, and then the body eventually kills the virus. My son Sean worked on a neat study on concussion. I'll show you that pretty quickly. He used that Deactor device to induce concussion, and what happens is after the concussion, you get a rupture of the blood-brain barrier, you get some devascularization, and you get, immune response, ends up damaging the cells. But he showed if you gave stem cells right at the same time of the concussion, the rats did fine. If they got placebo, it took 2 weeks to get back to the baseline. And this was true with both memory and motor skills tests. Then he did a study on 20 rats, and he approximated CTE by giving a concussion every week for 10. And what we see is here at the top are a bunch of rats who got expanded stem cells actually out of our lab, and below they got placebo. If you look at it closely, you'll see the MSC treated cells are almost pristine, the brain's almost pristine, and you can see the mottled effect from the placebo treated cells, placebo treated brains. It's a really great indication that if you give somebody stem cells right when they can get a concussion, you could probably prevent the CTE, and you probably can treat it too. Now remember, I showed you Dr. Jew at the start of the lecture. They do a really neat procedure where they fix this spine, and then they open up the spinal cord, and they clean off the scar tissue of the spinal cord. Now look at this video. I'll just show you part of it. This is from 9 days after a spinal cord injury. And what you're seeing looks like pus coming out of the spinal cord. But as you open up the fibers of the spinal cord, you'll get all this inflammatory fluid comes out. So what we're suggesting now is what you want to do is fix the spine, open up the spinal cord, get rid of the inflammatory fluid, and then treat it with stem cells. In fact, this is the inflammation you get. And quite simply, here was a patient that I saw back in March who was a C5-6 quadriplegic. This is Dr. Jew with her stem cells, the SVF, and here she's injecting it right into the spinal cord after the scar tissue has been cleaned off. Anybody who has a spinal cord injury beyond a week or two is not going to have that purulent or exudative fluid. They're going to have thick scar tissue. So the scar blocks the neurotransmission. But when we give the stem cells, we can regrow blood vessels and improve the neurotransmission. And take a look at this. This is that same patient 5 months later. She couldn't move her arms. Now she's standing and actually shifting her weight and moving her legs. She couldn't do any of this stuff before. This is what makes this so much fun. Now we're working with the American CryoStem company, with our company, Cells on Ice, and we're able to expand cells. We've done several validation studies that show that we can get safe, disease-free cells from the lab, and that these cells are indeed MSCs. Once we get the cells from the person as compressed fat, we send it back to the lab in this transport medium. The lab does a lot of quality control. When they get the cells, they validate sterility. When they process the cells at the end, they validate sterility. They'll take a sample 7 days later after they reprocess the cells and make sure it's still sterile. When we get the cells, we validate the sterility as well. And we've also validated that the cells have good flow cytometry. And this shows you that the cells, after you expand them, are no longer 34 and 45. But indeed, they're 73, 90, and 105, which are really the markers for mesenchymal stem cells. So once you expand the cells, you indeed have pure mesenchymal cells. Now, why all the confusion with the FDA? A lot of it started with a letter to the FDA that suggested, among other things, they said if we use lipoaspirate and digest it with collagenase, can we, and isolate stem cells, is this okay? And the FDA wrote back and said no. And said if you want to do that, you have to go through the FDA to get our approval. And so a lot of people think this means it's illegal to use collagenase. All it means is the FDA is not going to approve a letter like this because, first of all, they don't know anything about the collagenase, and the suggestion is you're going to make stem cells, and you would actually make stromal vascular fraction. And so the collagenase could have TSCs in it, could be, give you mad cow disease, and the FDA really has to police transmission disease. What people don't understand is the FDA was formed in 1938, but the FDA acts under the Public Health Service Act, Section 361, which was actually written in 1944 and gave the Surgeon General authority to prevent communicable disease transmission in foreigners. And that is the fundamental basis of what we work under right now, which is called 21 Code of Federal Regulations, Part 1271. So the whole purpose of the FDA rule and regulations prevent disease transmission. And the FDA has set up all these regulations, which basically suggest we have to go through the FDA to get any kind of approval. you really have to get their approval if you want to call what your product a drug. And so everybody has heard this pretty much, that the FDA will exempt you from going through their process if you're doing the same day surgeries, same surgical setting, using autologous, the same patient cells, for homologous use. Means I take out fat, but I have to use it as fat, and I can only minimally manipulate the cells. And we're not promoting our therapeutic results by making claims about our product. Fundamentally, we've known for all along we've more than minimally manipulated our cells, but that's based on the FDA's decision on how to define our cells. This is a little complicated because the FDA defines fat as a cushioning product, and we know fat is far more than a cushioning product. So even if you do liposuction, it's more than minimal manipulation. But fundamentally, we believe we're compliant with the FDA because we do everything to validate our production and show there's no risk of disease transmission. In fact, when you do a point-of-care procedure, you can't prove that it's not sterile because a patient could contaminate their own cells. When we produce the cells in the lab, though, we have a GMP facility that absolutely validates sterility from the start to the finish. And when we've done our transportation studies, we validated that the product we get is sterile by sending out samples to the lab to show that it remains sterile. So this is a good way to show that everything we're doing is compliant with the FDA. And on top of it, as I mentioned before, we filed an application with the FDA, so we'll do a knee arthritis study as soon as we get this application approved. And the other issue I think that's important is not only have we validated the sterility of our processed or expanded cells, we've also sent samples for flow cytometry, and we can show that the cells are indeed stem cells with proper identification, and they still grow out as they should. By the way, in case anybody's interested, the 261 strains of stem cells that were originally sent out by the government, NIH, to be studied, most of those were never validated for sterility. So technically, those cells could be, infected. We just don't know. In summary, first of all, thank you all for listening to me for the last 45, 50 minutes, I guess. I have a couple of questions to answer that we've written down. But I'm going to just say we're in a new era of medicine. There's no doubt about it. This is not experimental. It's really investigational. We've got years of safety data. And while we're seeing mostly chronic conditions, I really think this is ideal for point of care. And I think moving forward, we're really looking at this new era of cell renewal therapy and banking cells for bioinsurance. So where do we go from here? One little plug. We have a conference coming up. Any of you are interested, you can check us out on our website at either Stem Cell Revolution, Cell Surgical Network, or International Cell Surgical Society. It's our annual meeting of the Cell Surgical Network, June 2nd to 4th in Beverly Hills. Now, just real quickly before I end, and thank you all for taking the time, I had a couple questions I was asked to go over. And a couple of them were, if I want to do cryo collection, should I use my new cells or should I keep sending in cells to replace the older cells? Listen, your telomere shrinks as you replicate your cells. The cells in your fat, we don't even know how old they are because we don't know if they've been replicating or they've been sitting there dormant for a long time. And so we need to do studies on this. But fundamentally, it really doesn't matter. Think about it. If you have cells that are frozen as stem cells and you use those and they literally rebuild cells that are dying, what difference does it make if they live 10 years or 7 years? You're replacing old dying parts. And theoretically, if we do that, we should be able to extend our lives well into the hundreds, maybe 150 or 200 years. Who knows? Do expanded cells include any SVF? When we send cells to the bank, they process it as SVF, and then they take a sample of that, and when they expand it out as stem cells. And what's interesting is the way they expand it out, they can get multiple numbers, 50, 100 million of these stem cells multiple times. And we use a method of collecting the cells that doesn't use trypsinase, so we don't get the cells transfigured or conformed into fibroblast-looking cells, and they stay in their little round ball phase so they fly, right through the bloodstream, through the lungs and everything without getting stuck. I was asked, can expanded cells be shipped to my office in Florida? Right now, we're doing the first phase of our safety evaluation on expanded cells by working with everybody in the cell surgical network. Once that phase is done, we should be able to open this up to anybody harvesting fat, and then we should be able to provide you expanded cells. And hopefully we'll do it under IRB-approved protocols that cover anybody using expanded cells produced by our cell bank. Another question was, how do adipose mesenchymal cells become pluripotent stem cells similar to the embryonic? I'm not sure they do. A lot of people think that what makes a mesenchymal stem cell different than a pluripotent or induced pluripotent cell is that the DNA is wound so tightly you only expose the mesenchymal portion of it, and therefore the message only gets to that mesenchymal portion. We know from other studies that they can become more potent than that. As I said, Dr. C and Dennis Ling did a paper that you can read in the back of our book, and it's free to check out online. And it chronicles all these different things that SVF has turned into. So I think if SVF can turn into all these different parts, I'm talking about hepatocytes, myocardiocytes, renal cells, if it can turn into all these things, insulin-secreting cells, there's no doubt that MSC should be able to do the same thing. Somebody suggests they had bone marrow and adipose, and the bone marrow worked, but the adipose didn't work, and we want to know why, and it's better to use a mixture. What we're finding is a lot of doctors who were harvesting adipose were using tumescent anesthesia, and we tried this ourselves when we were in Hong Kong. We saw that if you use tumescent anesthesia and you leave it in a little bit too long, you can get a lot of cytotoxicity in a lot of patients. Also, there are some people, when you harvest the adipose, you can get low counts on the stem cells. We saw that when we did flow cytometry. But we believe that working with adipose works fine for the vast majority of cases. If it doesn't work, we tell people, come back, we'll treat you at cost, and we'll try to take the adipose cells from a different area where maybe we'll get a better cell count and a better result. And frankly, that happens quite frequently. And finally, we can always take the adipose and send it to the lab, and now we have the ability for the lab to grow the cells, and they'll give us exactly whatever number of cells we ask for. And do you do any type of testing on bank cells that you receive for injection to see if they're viable? Well, as I said before, we do two testing. First, we make sure the cells are clean. The patient, first of all, is tested for infectious disease. You can't even begin to bank cells unless you clear them of hepatitis, HIV, and a few other communicable diseases. Once you do that, the bank checks for infectious disease and then processes cells, and they do a quality control sample that they can test for viability by showing that the cells grow out. And and before the cells are ever released, they're also cleared for infectious disease. And when they're processed for release, and what the neat thing is, our bank can process the cells and send them back, so you just take them out of a sterile vial, and you don't have to thaw them out or process them. Anyway, I think that covers our time for today. I want to thank you all for joining me. It's really been a privilege for me to speak with all of you. Thank you very much.